Hey, Bio2 students, Mr. Yantz. Starting a new chapter today, chapter 7, which is really kind of a continuation of the last chapter. So the cell, we're just going to focus in on cell membranes and not just their structure, but how things flow through them, diffusion, osmosis, things like that. Okay, so let's start with their structures. You see on the screen a cell membrane. It's hard to see in our microscope. You're just seeing that border. If you really break a cell membrane down, it's got lots of stuff in it. It's got carbohydrates, it's got proteins, it's got fats, it's got all kinds of things embedded in it, and that's what we're going to cholesterol. We're going to talk about a lot of different things in this chapter. So let's just jump right in. So if you look at a cell membrane, it's pretty amazing how thin it is. It literally is 8 nanometers, which means it would take 8,000, think about that, 8,000 cell membranes stacked on top of each other to make one page of a book. Isn't that crazy? One page. So they're extremely thin. However, they're obviously very important because they are selectively permeable. It's a good old Bio 1 concept we talked about back in the day, which means that they're not permeable, which means they don't allow everything in, and they're not impermeable, which means they let nothing in. They're kind of in between. So how they do that, again, as I say hundreds of times, structure determines function. So you're going to find lipids and proteins. That's the two main ingredients of a cell membrane, fats and proteins. So what you see with that is something called amphipathic. Amphipathic means I got some conflicting things going on the outside and on the inside. So think of a membrane having a top layer and a bottom layer, and then you got this stuff in the middle. It kind of interesting. The outside and inside are what we call hydrophilic. So they have these hydrophilic heads, which means a love of water. And then they got these tails that stick down and up. I'll show you a picture in a minute. Those are hydrophobic, which you guys probably know. A hydrophobe is someone who fears water, right? So you see we have conflicting things. You can see it down here in this bottom picture. We got these hydrophilic heads that love water, bringing water to them, both inside and outside the cell. And then we got these tails that stick down, and they're hydrophobic, and they repel water. So if you look closely, you'll see little white lanes in there. We'll do this more later. So water can flow between those hydrophobic heads and also a little bit laterally, but we have those in there so things just don't go everywhere, okay? We don't want a floodgate. We don't want to just open the cell membrane up and say, just everything come in, because there's some stuff like, bacteria and things like that, viruses. We don't want them coming in, right? So that's kind of what makes them amphipathic, is we got both hydrophilic and hydrophobic working together to make that membrane selectively permeable, which makes sense if you think about it, okay? Now, in 1972, a long time ago, there was two famous scientists in cell studies called Singer and Nicholson. They won the Nobel Prize in science, and they came up with this theory that maybe a cell membrane isn't quite a static fixed structure. We thought that for a long time, that it was this rigid fixed structure that things flowed through, and then they go, no, it's more like a mosaic. I don't know if you know that from art class, where you take different like tiles or things and you put it in this design. It's sort of like you got these pieces all over the place, these proteins, and then we got this lipid layer that has a little flex to it, and that's really what a cell membrane is. It's this, there's Singer and Nicholson, it's this fluid mosaic, and you can see it's got a little wave to it. Cell membranes have some give to it. If you watch the video, I'm not going to play it right now, but if you watch the video later, I'll put it on later, you can see what I'm talking about. So it kind of changed our theory of what cell membranes really are. And then further investigations and electron microscopes proved that, oh yeah, this is a conglomerate, a mosaic of all kinds of stuff. Look at all the stuff on this picture. Glycolipids, carbohydrates, glycoproteins, cholesterol, integral proteins, peripheral proteins, cytoskeletons. There's stuff everywhere in the cell membrane. And it all has a purpose. And I think that's kind of what I want to emphasize today. I'm not going to give you a lot of notes today, just kind of want to introduce it. But the cell membrane, these phospholipids in the cell membrane, that's what gives membranes fluidity. It allows them, they're not a static sheet. They're not a fixed sheet. They have some give to them. Things can pass through them. And things don't move laterally a lot, but there is some flip-flopping that occurs. We'll talk about that. And cholesterol, we think of cholesterol being bad, and it is, some cholesterol is. But in this case, it's really important we have this cholesterol in there. And here's why. Warm temperatures. So there's a reason our body temperature is set at 98.6. And at that temperature, cholesterol allows membranes to be fluid, have a little give to it, and stuff can pass through. At lower temperatures, they get compacted. Okay, so if someone's has hypothermia and their body temperature drops really low, things don't move through the cell membranes very well. If you have a really high temperature, 102-degree fever, you don't feel very well. Things aren't functioning well because things aren't moving through cell membranes very well. So I think you have to understand the importance of a cell membrane. Just to give you a little personal story, my uncle's been in the hospital and he's having some issues in his body. And one of the issues he has, talking about cell membranes, is not trying to be gross or disgusting, but his bladder was not passing urine through those membranes and going out of the body. And when we put him in the hospital, they took out, think about this, a 2-liter pop bottle full of urine out of his bladder. It's that much that was full because these cell membranes, for whatever reason, we don't know why yet, we're still trying to figure that out, it wasn't getting rid of that like it's supposed to, and then that makes you feel bad because it kind of poisons you inside. So that's kind of a real life example. I think sometimes we get hung up on, this is the most exciting chapter. This is kind of boring stuff today. But think about the importance of how things have to move in our lungs. We have to move O2 and CO2 through our lung cell membranes. We have to move stuff through our intestine. We have to move stuff through, the liver, the kidney, the bladder, the heart. And we got to have this stuff working correctly. So we're going to kind of dive into this a little bit more. I mainly want to focus on how things flow through there, like osmosis and diffusion. We got a lab we're going to do on that Monday. So hopefully you'll see that in real time. All right. Hey, have a good day, and we will talk to you later. See ya. Bye.